Okay, this is Microbiology Lecture 17. Now, last time we spoke about the innate immune response or innate immunity, which is a non-specific response, and now we're going to talk about adaptive immunity or the adaptive immune response, which is a specific, very specific immune response. This is mediated by T lymphocytes and B lymphocytes, which often are called T cells and B cells. Okay. So the adaptive immune system or adaptive immune response is mediated by B lymphocytes and T lymphocytes, or B cells and T cells. Which means the same. Now, these cells, as they are produced in the bone marrow, like any other blood cell, they come out of the bone marrow. Each one has on its surface a receptor for foreign molecules. And each different one has a different shaped receptor for a different foreign molecule. These foreign non-self molecules are called antigens or target antigens. I'll try to use the term target antigen more often just to emphasize the fact that it's what the adaptive immune response is responding to. So each B cell has on its surface a receptor for a foreign antigen. And in the case of the B cell, those are antibody molecules. In the case of a T cell or T lymphocyte, it's the T cell receptor. And each different B cell or each different T cell that's produced, it's a individual clone that gets produced, will have a unique receptor for foreign target antigens on its surface. These foreign target antigens are anything that is non-self. Now, there are billions and billions of molecules that are non-self that could get into you, and some of them are on microorganisms. So you have in you, among your billions of T cells and B cells, you have a few cells that can, a small number, but nevertheless you do have some cells that can respond to those foreign molecules because they have the right target antigen receptor on their surface. So antigens are foreign non-self molecules, and you have B cells and T cells that have antigen receptors. In the case of the B cell, the antigen receptor is an antibody molecule on the surface. And in the case of the T cell, it's a T cell receptor. If a B cell or a T cell meets up with its target antigen, it gets activated. And a B cell or T cell that is activated will start to divide. And the cells divide rapidly. You know, they're doubling, say, every 12 to 24 hours. And that may not seem like much if you only have a small number, but when you get doubling, after 10 days roughly, you have a very large number of cells, and they're all identical, and they all have the right receptor for the thing that got into you. The other thing about activation by contacting or binding to the foreign target antigen is that in the case of B cells, not only do they divide, but they secrete their antibody molecule, their antigen receptor molecule, so that it goes throughout your body. T cells don't do that, but they do divide. So these are very, very specific responses which take time to occur. That's why they have the name adaptive. They'll adapt to the presence of that foreign molecule in you. If the foreign molecule is it's inanimate, it's not alive in any way, it's not a microorganism, then usually it gets cleared very early by the innate immune system response, just phagocytosed and, destroyed that way. It's not increasing in its numbers, and it's dealt with usually fairly quickly, unless there's an awful lot of it somehow that got into you. You don't really, trigger any specific response. But if it's something associated with a microorganism, and that microorganism is growing and doubling, if it's a bacteria, for example, it can double even up every 20 minutes. So there's a battle going on between your innate, non-specific immune response and the thing that gets into you. And if that thing manages to evade, the innate system and is still around and is even growing, and the infection is active, then you have enough time to respond in terms of your adaptive immune response, and you start having the right B cell or T cell or both multiplying and becoming large enough in numbers to have an effect. I'm going to talk quite a bit about antibody molecules, in other words, the B cell response, more so than the T cell response in the next few slides. People are more familiar with the idea of antibody molecules, and I will talk some about T cells, and there are many parallels between the two, but the main difference, of course, is that B cells secrete their antigen receptor, the antibody molecule, whereas T cells don't. So antibodies, which I want to point out to you, do start with the same three letters as antigen, but are completely and totally different. Do not confuse the two, which many students do. Do not confuse antibody and antigen. Make very, very sure that you understand what an antigen is and what an antibody molecule is. The abbreviation, by the way, for antigen is AG, and for antibody is AB. Now these molecules, antibody molecules, are proteins, and they're produced by B cells. They're on the surface of the B cell. And in response to binding of the antibody molecule on a B cell to its specific target antigen, the B cells will multiply. Those particular B cells with that particular antibody will multiply and start secreting the antibody molecules. Now the whole idea here is that the antibody molecule binds specifically to the antigen. The same idea actually for the T cell and its receptor, which is called the T cell receptor. It binds specifically to its target antigen. So activation involves binding, and that's the whole role, and that's pretty much it between antibody and antigen, or T cell receptor and antigen. Binding. After that, other responses are triggered. The B cell mediated immune response, which is, essentially antibody production, has in the past, of course, been called antibody mediated immunity, and an even older name is humoral immunity. So you may see that still, or hear it, humoral immunity. The T cell response, or T cell mediated immune response, is called cell mediated. Here, humoral actually means a fluid. And so the idea is that it's secreted and released, whereas here the response is directly associated with the cell and stays with the cell. Now, antigens are foreign molecules. The thing that gets actually recognized on the foreign molecule is called an epitope. That's the specific thing that gets recognized. These are also called antigenic determinants, but you may hear the term epitope. That's what is recognized. Here, in this case, you see it's not the bacteria, the whole bacteria. That would be a huge. And in fact, the sizes here of these antibody molecules has been exaggerated. They're much smaller than this, but they're exaggerated in this diagram. But the idea is that a microorganism like a bacteria will have several different epitopes or antigenic determinants on its surface, not just one. There are many molecules, in fact, on its surface that are foreign to you and could trigger an antibody response. And those antibodies, their shape is the overall molecule, which is a fairly complex molecule. The overall shape looks kind of like a Y, but it's made up of 4 polypeptide chains. And the binding sites, where they, remember the whole role of this, molecule is as a receptor for foreign target antigen. So it has to have a binding site where it interacts with the antigen. And the binding sites are at the ends of the arms right here. Okay, and that's shown here where this antibody is binding to that particular antigen or antigenic determinant, or better, the best name is actually epitope. And here this is a different antibody molecule. Antibody molecules in general have 2 binding sites, so here it's showing both of them binding to the exact same target antigen, right? But this antibody molecule here could have bound also onto here or here or here and here, right? Those 2. So foreign molecules are antigenic determinants or epitopes. A microorganism, which is much bigger than a single molecule, I mean, it's made of, it's giant in terms of molecules that make it up, has many different potential antigens on its surface. Sometimes, I don't know if I want to teach you this so much. It's good for your own information. The idea is that here that sometimes you have foreign molecules that are too small to trigger an immune response, but if they get combined with what's called a carrier molecule, then they can trigger an adaptive immune response. They could trigger an antibody response or a T cell response. They're just too small on their own, but when they're linked to a carrier molecule, then they are arranged in such a way that they will, they're arranged sort of in a linear or associated pattern and are big enough now to trigger a response. And these things are called haptens. Foreign molecules that by themselves can't act as an antigen, but when combined with a carrier can. And actually a lot of vaccines, or a number of vaccines, I believe that involve carrier molecule and haptens coming from the organism, hapten molecules plus a carrier, and you get a good response. Okay, just in terms of, and these, I got to tell you, you won't, they're sort of older, more very general terms. You do run into them certainly, and it's worth understanding. In your plasma, remember that plasma is the fluid part of unclotted blood, but once it gets clotted, the liquid remaining is serum. So serum is essentially plasma without clotting factors, right? Once it's clotted, all the clotting factor proteins are used up, and it's serum. Now, your serum or plasma contains many different antibody molecules, which are there because of different foreign target antigens that got into you and that triggered an immune response, and those molecules are still there. So you have a whole mix of different antibody molecules in your serum, and if you collect the serum from a person, and if it has a particular activity you're interested in, it's called an antiserum. Antisera usually are not used for any treatment because of the risk involved in other types of infections that might in that way be transferred over to the recipient. But they do use sometimes immune antiserum from someone who's immune to a particular thing. I'm trying to think of a good example. They may use that for like snake venom antiserum, collecting it from a person who's been exposed to it. They do that with that type of treatment has been done for COVID-19 infections, people who have recovered from the infection with SARS-CoV-2, which is the coronavirus type that causes COVID-19. They use that to treat individuals. So antisera just means the serum containing antibodies. Serology is the study of reactions between sera and target antigens. Globulins are the serum protein, the class of serum. There are many globulin proteins. These are globular proteins essentially in your plasma or serum. There are many different kinds. The one subgroup contain all the antibodies. All the antibody molecules are found among the gamma globulins. And if you take a person's serum and run it out on you do a serum electrophoresis, it's called, where you put it in a gel and you apply a current. So you attach the negative here and the current is flowing in this direction. If you put your sample in the trough here, the serum sample, and apply this electrical field, then they will migrate this way. They get pushed forward and they migrate according to their size characteristic, and they'll band out. And among all the globular proteins or globulins in your body, the gamma globulins contain the antibody, and they'll be here. That's what's meant by gamma globulin. It's simply a subfraction of your serum. Some people sometimes talk about gamma globulins. They're talking about a mix of different antibodies found in the serum. Here's albumin. As you know, albumin is the most commonly found protein in the plasma. It makes up about 50% of the plasma proteins. It is important in fluid balance and maintains the proper osmotic pressure between the tissues and the plasma. Okay, let's talk more about antibody molecules. As I told you, antibody molecules are made of 4 polypeptide chains. 2 of them are called heavy chains, and 2 of them are called light chains, for the simple reason that 2 of them are bigger, the heavy ones, than the other 2, which are the light ones. The 2 heavy chains are identical, and the 2 light chains are identical. And here you see the arrangement in a stick figure kind of drawing of the heavy chains. There's one of them is on that side and the other one's on this side in green, and the light chains in blue. And these 4 chains are held together by disulfide bonds arranged like this. And I should point out to you that this area of the molecule in here, right in there, is called the hinge region. And it's called the hinge region for a reason. It's because it's like a hinge on a door. It allows it to swing. So the two arms can swing. They can look like this, or they could swing more open like that, or they could swing more closed. They are quite flexible because of that hinge region present right in here, in that area, the hinge region, just like on the hinge of a door. As I've told you, antibody molecules, their job is to bind a particular specific foreign target antigen. And the binding site, there are two binding sites, and the binding sites are here and here, the antigen binding site. So there are two antigen binding sites. And it's very much a very specific type of binding based on the shape. This stick figure does not give, a clear idea at all about the fact that the end part of the two arms there, which is called the variable region, it has a very specific kind of shape and varies from one B cell to the next. All the antibody molecules on a single B cell that was produced out of your bone marrow will have a particular shape at the end of the arms. And that can bind because it fits nicely physically to an epitope, an antigen terminant on a foreign molecule. And here you see a pocket, the kind of pocket that you would get in here between the two arms, in the space here, you see, and there's the antigen binding site. They're showing part of the heavy chain, the variable end of the heavy chain, and also the end of the light chain. And they're showing that this determinant, this antigen, fits in nicely, perfectly into that space. And so you get binding of a particular target. And that occurs in those two regions, there and there. Now each chain of the four chains, the two light chains and the two heavy chains, they have what's called a constant region. There's a constant region, there's an identical constant region, and on any other antibody molecule of this class, it doesn't matter the antigen specificity, they're constant. And on the heavy chain, also a constant region here and here. The specificity for a particular target antigen is not found in the constant region because all the antibody molecules have similar constant regions. It's found in the variable region. The variable regions on the antibody molecules on one B cell will be different from the variable regions on the antibody molecules that are on the surface of a different B cell. And that's why they have different target antigens. Each B cell has a different target antigen because the antigen binding site made by those two variable regions of the heavy and the light chain, this area in here, is unique to that particular B cell and its antibodies as compared to the next B cell and its antibodies. And this is identical here also, and so there are two antigen binding sites that can bind these epitopes or antigenic determinants found on a foreign molecule, foreign antigen molecule. Okay, so this is the structure of a secreted antibody molecule. B cells have these embedded on their surface. This is the end of the molecule that's embedded in the membrane of the B cell. And these molecules are sticking out with their arms, their antigenic binding sites sticking out on the surface, all over the surface of the B cell, just waiting for the right foreign antigen to come along for them to bind to. And if they do bind it, then the cell gets activated and starts to divide and starts to secrete the antibody molecule. Here's a different kind of drawing, same molecule, same idea, but a slightly different drawing where you have the light chain here, identical light chain here. In yellow, you have in green, the heavy chain here, and another heavy chain right here. And you see the disulfide bonds in there, holding the two heavy chains together, and the disulfide bonds there, holding the heavy and light chains together. And this, of course, is the, let me just get rid of some of this. This is the hinge region that allows the two arms to swing, right? You see the variable heavy, variable heavy, variable light regions. There's the antigen binding site, very specific, identical to each other on one molecule, but different on the next B cell. Different kind of molecule showing all these things that I mentioned. Variable light here, abbreviated VL, variable heavy, variable light, variable heavy. Remember, the two light chains are identical, the two heavy chains are identical. Each one has a variable region, each one has a constant region. The constant region, of course, on the heavy chain is much longer. There are the two binding sites. Okay. This kind of rectangular representation of the variable light, this area of the protein, remember this is a protein chain, this light chain, that's a protein chain, the heavy chain. It's a polypeptide, right? So these areas are called domains. There are two domains, a variable light domain and a constant light domain. On the heavy chain, there are 4 domains, 1 variable and 3 constants. So, you hopefully understand that you produce huge, gigantic numbers of cells in your bone marrow. Many of them are red cells. Fewer, but still many, are white cells, and among those white cells are, all the different lineages, the different types that end up being either myeloid or lymphoid, or some become thrombocytes. But the lymphoid are the ones that form the adaptive immune response, the B lymphocytes and the T lymphocytes, or B cells, T cells. And you hopefully understand that you actually do produce billions of B cells, each one unique out of your bone marrow, each one with different antigen specificity, a different target antigen that it can bind to. When you look at those antibody molecules, even though they may have every single one has a different, particular antigen binding site, nevertheless, there are classes that they can be grouped into. And for antibody molecules, there are 5 classes, 5 different classes. They differ from each other quite a bit, although the overall idea is the same, that they can bind to a specific target antigen. But you have 5 different classes of antibodies. And I just always call it GMADE. GMADE. But you have 5, IgG, IgM, IgA, IgD, and IgE, GMADE. Ig stands for immunoglobulin. Remember, it's immune. It's a globulin, which is a globular protein in the plasma. And there are 5 classes, G, M, A, D, and E, and we'll go through each one and talk about some basic fundamental characteristics of each. First of all, when you look at the 5 different classes, these molecules have a number of repetitive subunits. The one I described before, that's called a monomer. It has a light chain, a heavy chain, another heavy chain, and a light chain, a single monomer. IgG and IgD and IgE are monomers. We're not going to talk a lot about D. There isn't exactly a huge amount known, and it doesn't seem to play that big a role, it seems. IgG and IgE we will talk about. IgA are dimers, and you'll see what I mean later. They're actually 2 of these structures linked together. And IgM, here we talked about IgA. IgM is a pentamer, so that there are 5 of these all linked together on these molecules. Pentamer. In terms of, if you look at the antiserum, the serum antibodies, if you look at all the serum antibodies, as you know, they're gamma globulins, you find that 80% of them are IgG class. So IgG is the most common class of antibody molecule found. IgA are important. They are found in the serum. They are also found secreted out of the body onto the surface of mucous membranes. So you'll find them in the respiratory tract, in the digestive tract, in the genitourinary tract, in breast milk. Very important. So G is the largest component of all the IgG class, then IgA, and then IgM. In terms of how long, these are protein molecules. They don't hang around forever. B cells are producing them and secreting them. The B cell lives for quite a while. After a while, after some time, it will go into sort of a resting phase where it's not multiplying. If the antigen it's specific to is gone, all the B cells don't die off. They hang around, but they won't be secreting anymore. But initially, with exposure to a foreign molecule, whatever B cell can respond to that will start dividing and secreting antibody, and you'll have it in your serum. The antibody molecules are protein molecules. They have a half-life. In other words, half will be gone by 23 days. That's for IgG. So the longest lived is IgG, and the others are much shorter. This is an artist's diagram showing the overall molecule, but remember it's got 4 polypeptide chains that are held together by disulfide bonds. Occasionally in books, most of the time you'll see it like this, but sometimes you'll see it drawn like this. There is no difference. You can get rotation around the hinge region that ends up looking like this. So don't get confused. There is no difference. It's not like one is wrong or you thought it was different. It is the same molecule. Now, IgG class molecules, they have the ability to activate or fix complement by the classical pathway. Remember the classical pathway, C1, 4, 2, 3, 5, 6, 7, 8, 9. 1, 4, 2, 3, 5, 6, 7, 8, 9. Classical pathway. They typically are, that's an important fact that they can fix complement. Not every class of antibody can fix complement. IgG class antibodies are found in the blood, in the lymph, in the wall of the intestine inside, not in the space. Okay, in the blood, in the lymph, in general throughout the body. Intestine is a bit much actually, but now IgG class molecules also, another important feature are that they can cross the placenta. Not every type of antibody can cross the placenta and go into the developing fetus, but IgG can, and in that way they protect the fetus, because the fetus's immune system does not develop until after birth, and it's during the first 6 months that it's developing. It's not completely developed until 6 months of age, although newborns are vaccinated, so their immune system is triggered. They receive vaccines at 2 months and 4 months. You know, the response is not complete always because it's still developing, but it's important to protect them early on. But the immune system doesn't actually fully develop until 6 months of age, which is okay to get vaccinated, because the vaccine is not going to make you sick with the microorganism. It doesn't contain the live microorganism that could actively replicate and cause an infection. You're just getting exposed to the foreign antigens, that's all, just to trigger an immune response. Anyhow, the idea is that in the fetus doesn't have a specific immune system, so the antibody molecules made by the mother will cross into the fetus and protect the fetus. So antibody molecules of the IgG class can cross the placenta and protect the fetus. They do a number of things, including they'll enhance they'll enhance phagocytosis if they bind onto the surface of anything. They can also act to neutralize toxins and viruses. Remember, the half-life is 23 days. It's a monomer and makes up 80% of the serum antibody. IgM class are pentamers. That means there's 5 of those 4-chain molecules linked together. They also fix complement. They are also found in the blood and lymph, floating around. They help to agglutinate microbes. They help to bind together several microbes, which makes them more susceptible to cells like phagocytic cells to bind, attach, kill, phagocytose and kill. Whenever you get exposed to a foreign antigenic molecule, the very initial first response is always an IgM class antibody. So it's the first type of antibody produced in response to an infection. Again, this is just a repeat of what we said earlier. IgA class antibodies are dimers. There are 2. So IgG is a monomer, IgM is a pentamer, IgA is a dimer. They are found in secretions in the mucus, and remember, mucus is found in mucous epithelium, and mucous epithelium are in your respiratory tract, genitourinary tract, and digestive tract, and they are also found in mother's milk. And one thing that's missing here is sweat, perspiration. They'll be found sometimes on the skin surface. Okay, they provide mucosal protection. I'm not sure about that. Let me think. I'll have to look that over. But certainly in mother's milk and mucus secretions. So they protect the mucus, and they're in mother's milk, particularly the very earliest. And so they protect the fetus, because the fetus, not the fetus, the newborn. Fetus does not breastfeed, the newborn does. Right? So they will also protect the newborn who is breastfed. Breast milk is something that has evolved to nourish and protect newborn babies over the past 100 to 500,000 years, and is way, 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 way better than formula because of its complete range of nourishment, but mainly because of IgA class antibodies that are found in that breast milk. Okay, so the next one should be IgE. No, it's IgD. I'm not going to say much about this. It is a monomer. I'm going to leave it alone. It's found on early B cells that are as they first get produced. I'm not going to get into it. IgE antibodies are monomers. They are able to bind via this end of the antibody molecule. They can bind. When they get secreted, they can bind onto the surface of mast cells and basophils. Remember that basophils are found in the bloodstream, and if they leave the bloodstream and go to the tissue, these are a type of granulocyte. When basophils leave the bloodstream and go into the tissues, they are then called mast cells. Mast cells and basophils, if I were to draw it like that, have on their surface receptors for this portion of IgE molecules. That part of the IgE, or that part of any of these antibody molecules, is called the Fc receptor. Did I say receptor? The Fc portion. And what basophils and mast cells have are receptors for IgE Fc. So IgE class antibodies, the Fc portion, that's the tail end here, in this area here. Actually, all of that of an IgE molecule is the IgE Fc, and basophils and mast cells have IgE Fc receptors on their surface. Okay, so they can bind these antibodies by that tail end, by the Fc portion, and then the cells will have all kinds of different IgE class antibodies on their surface, sticking out like that, sticking out their arms that have, an antigen binding capability. It could be these are different antibody molecules because they were each maybe secreted by a different B cell. Okay, so they're all over the surface. These IgE class antibodies are typically mostly involved in reactions to parasitic worms. So you tend to find them in larger numbers in people who have parasitic. We, after all, evolved in, constantly endemically infected with parasitic worms. Most of the world's population is, in fact, infected with parasitic worms. They're also, unfortunately, involved in some allergic reactions, these IgE class molecules, antibody molecules. Okay, this is the end of Lecture 17, Part 1.